first. So I gave this talk, basically, I try to promote the idea about how we can do combined studies. We're using imaging as we all do here, but also we can combine different level of information to enhance our understanding. First, my talk is about schizophrenia. As you all know, that schizophrenia is genetic disorders, and heritability estimated to be 70 to 80%. In recent years, there's a large study, collaborative, the whole world, have produced a very promising, very good quality studies, produced very high profile of risks, genetic risks for schizophrenia. Since then, we can say a shift from the researcher, which shift from the association study, produce the risk, to causations. Try to understand what is the mechanisms under these risks. We all know from understand the mechanism underlying is not an easy task, and there has been a variety of ways to enhance our understandings. I should give example what other ways we can use, like understanding. Okay, so probably you can say in vitro can use traditional cell lines or iPSCs induce pluripotent stem cells and iPSC induce neurons. All these cell lines can produce direct information about genetic mutations induced cellular change. You can say all the transcriptome change, protein change, cell formatry changes. And the other modality you can use is postmortem brain tissues. And studies have used collecting the postmortem brain tissues from fetus to adulthood. Then you can see all the transcriptome cellular level, there is cell morphometry levels and brain organ levels change. But all these are done basically postmortem or in lab. In vivo, what we use is like everyone here using the live brain. We can use imaging or signal techniques to mirror that. And in company with that is peripheral tissues, which we can collect relatively easily, use like blood or salivas. You can see the transcriptome, protein, or cellular level changes. The point I want to make is all this information we derive from here are not independent. They complement each other. And there are some examples we can show here, but I will not go into details. But all this study promote is we want to have integrated studies. Then we can combine the information together. And then that can increase our knowledge about the whole pathway, not the point by point. So the following I will show is 3 of our studies working towards that direction. The first is cross-tissue genetic epigenetics study. So in this study, we have patients with schizophrenia and have the controls, we have discovery set and the replication set. The data we analyze is the SNPs, which in the schizophrenia risk region was reported by the large study in the Nature papers. Then we add a different level of that. We add the DNA methylation collected from blood as blood and saliva. One is discovery, one is replication. And also we have collecting the same people, the live brain, looking at the brain's imagings. And we also have assessed their cognitive and symptoms. Then in this study, the first result we have is we can see clearly very significant SNP, the genetics relation with DNA methylation. DNA methylation is one of the major epigenetics with regulating gene expressions. So the example I can show here is in the chromosome 10 region. In chromosome 10 region, about 1 million base pairs cover several genes. Some of them are already reported related to schizophrenia. And the SNPs here working collectively influence the DNA methylation of the CPG side. And the loading of the DNA methylation is significantly related to the loading of the SNPs. Using one example, you can say this is one SNP, this is one CPG site. You can see the number of minor alleles is significantly modulating the level of DNA methylation. This relation between genetics and the DNA methylations are consistent in blood and saliva and also has been reported previously in postmortem brain tissues. sorry, I was messed up here. So then secondly, we're looking at is the relationship between DNA methylation structure, brain structure, and cognitions. So in this results, we showed is, you can say the DNA methylation component here, mainly in the NOSEP gene, was regulating nitric oxidase activities related to the 8 of the green matter components here. And a schizophrenia patient has hypomethylations, and at the same time, patient has reduced green matters in 7 of these brain components, significantly in the frontal region and temporal regions. And 5 of these brain components also related to the cognition, working memory, and the problem solving, which is consistent with reduced green matters with poor cognition. And in our replication data, 5 of these pairs are replicated between DNA methylation and the brain, and between brain and the cognition, 4 of them are replicated. It's quite consistent relationship. Another point I want to make is one of the DNA methylation components shows promising mediation effect. So the same region, the DNA methylation, mediated genetic effect to the brain, which put them together, gave us the summary is the periphery methylation patterns had related to the brain, and we can explain the differences of the brain and cognition in patient. Then that leads to the questions how. That there are 2 possible mechanisms for that. The first one is the same environmental insult can induce the DNA methylation chains across multiple tissues. For that, the animal study has proved the possibility. The second mechanism is the same genetic mutations also can regulate DNA methylation across multiple tissue types, which is basically cross-tissue, the MEQTL. Then that leads to our next study is we want to understand systemically what is the cross-tissue MEQTL's properties and how that relates to psychiatric disorders. So in this study, we have we collect the data from 3 tissue types: postmortem brain, blood, and saliva. And data from brain and saliva are come from brain from and blood are come from published results. So very great for the authors to give us very detailed results, and saliva is from a local lab. In this result, we showed is that there are quite amount of MEQTL in genome-wide in each of the tissues. In each of the tissue, brain, blood, and saliva, and among them, you can see the 45 to 70% of that are shared MEQTLs, and also 30 to 60%, 68% of the target CPGs are shared across tissue types. So quite amount of the common things there. And within the shared, which we call the cross-tissue MEQTLs, that 80 to 90% of the same direction, same effect direction, means that if the minor A-level SNPs increase DNA methylation, then the effect is consistent across tissue types. And the ranked correlation of the effect size among 3 tissues is quite also very high, it's 0.8 somewhere. This information more interestingly combined is that we're looking at the CPGs which targeted by the MEQTLs has a greatly increased chance to show very strong blood-brain correspondence. Here, the blood-brain correspondence defined as the methylation we observed in blood can explain more than 50% of the variance in brain. So it's quite explain large of the variations there. And when the effect size is higher, the probability get higher. So if your MEQTL effect is large enough, which is guaranteed, the methylation in blood and brain are correspondent. Then we also look at the cross-tissue MEQTL, the SNPs. They are significantly enriched for the multiple diseases, and particularly for schizophrenia. Then that leads to our third study, which is we have compiled the SNP, DNA methylation, expression, and the gray matter. So in these studies, we have discovery and replication 2 data set, quite large of the sample, and we focused on the 1,400 SNPs, which reported for significant risk for schizophrenia, and we looked at the whole brain gray matter. So first, in these results, we can say is we identify one brain region, which is temporoparental junction areas. The reduced green matter of this region is significantly related to a particular set of genetic risk. This genetic risk is combined of 39 SNPs in the chromosome 6 P22.1 regions. And in this connection is also replicated. So that is showing the green matter and genetic risk connections. And further, we want to emphasize here is we're looking at these SNPs. What is the regulation effect? So we can say in this plot, 31 of the SNPs has shown the regulation of DNA methylation in postmortem brains. And the postmortem brain, the DNA methylation in patient has shown the hypermethylations. And also, we say that one of the SNPs regulating DNA methylation consistently in blood and in brain and saliva. So that gave us a chance to looking at in vivo the methylation in saliva, how they relate to the brain structures. Oops, that's kind of behind a little bit here. So you can see from here, we're looking at the in vivo, the same population. We're looking at their saliva, the DNA methylation, and their brain's brain matter reduction, which indeed show the significant correlation here. And further, we're looking at the same SNPs. They also regulate the gene expression of the postmortem brains. And then we can see in postmortem brains, the patient showed the downregulation of the gene expression. Overall, we can see the risk alleles we discovered from reduce the green matter using imaging data are all consistent with the risk alleles we reported for the risk for schizophrenia in postmortem tissues. So if you're looking at that, that gave you information, a possible way for these risks to exit its effect. So it goes through the DNA methylation, goes through the gene expression, and then that provided a pathway to understand this underlying mechanism. So what is the future directions? Here I try to promote is that I think the future should focus on the integrated studies. And we can leverage all the information we can get to put a whole picture together. And here's one example. For example, we can use, right now, the iPSA-induced neurons can be, it's not popular, but it's coming up so quickly. We can use this to understand exactly what is the molecular features that genetic risk of mutations can give. When we have these molecular features, then we can look at using postmortem brain to looking at which regions have expressed such strongly the molecular features. If we locate what's the molecular feature you want to use and what the brain region you want to monitor, then we can use involved information here. We can looking at the brain imaging or signals they collected, and we can use the molecular features that we use in peripheral tissues. Then at the same time, we can monitor the trajectory of the brain changes as well as the molecular feature changes. Then that can give us more information of the whole picture, how the disease developmental trajectory. So all this work cannot be done with a large collaborative effort. And here we only list of the representative of each groups. And in Mia Lab, and Dr. Vince Cajon, and also the Dr. Jiayu Chen and Dongdong Lin, they are basically the first author of the papers we presented here, and also collaborated with the professors in UNM and professors Jessica Turner in Georgia State. Okay, I did use my time. Okay, good. Thanks.